the second session for nephropath guidance, I guess is a great way to say it. I'm looking at people who have started to join in. Everybody should be able to see my screen. If you cannot see the screen, then please raise your hand with this. Well, I guess you couldn't see my screen, so I can't show you where the little raise my hand button is. But if you're up in the participants window, you should be able to click on raise your hand, and then you could either unmute yourself or you could type in a question if you have any questions. And it looks like Dr. Sanchez just joined us and Dr. Schneider did. They are both unmuted, so anything that they say will be recorded, which is fine. Oh, now they're muted, okay. And are we recording this session yet, Mary? Almost. Or can I record?
Yes, we are.
Okay, cool. So, I'm just gonna go into presentation mode. So we already did the first 3 myths. I only have 2 more myths, but they're the most exciting of the myths. And so the first one is that I do not like H&E. I think, especially for kidney, H&E is almost virtually worthless. And you guys have heard me say this if you've interacted with me in any way, shape, or form. I use H&E only when I am trying to figure out what type of inflammatory cell type it might be. But I can usually tell that on other stains, but I really don't think H&E is a good stain. So we're going to talk a little bit about what stains can be used for kidney and how we might want to, and what they should label and how they should stain things. So yeah, H&E is good. Maybe if there's a tumor, maybe if there's papillary necrosis, because all you see is pallor in that region. But in general, I don't think H&E is a good stain. And this little hi here was Mary and I practicing annotating on slides. So apparently I can start to draw on my PowerPoint slides. Okay, so what special stains should we use? And the first one is the periodic acid Schiff. That's probably the go-to one that we learned about when we were doing the WSAVA study. It was the first, oh my gosh, the high stays there. I got to get rid of that high. Can I get rid of it? Clear. All drawing. Yay. Okay, I learned. So PAS is the first thing we would look at. So the PAS is going to help us evaluate the glomerular basement membrane. It's also going to help us evaluate the mesangial matrix. Please realize that the GBM and the mesangium should be the same color on this stain. They're also the same color usually on the H&E, but if you're looking at a PAS stain and the GBM is a different color than the mesangium or vice versa, then you have a disease process going on. Usually the situation would be that the mesangial matrix is a different color than the GBM, and that would be suggestive of maybe amyloid deposits. So if they're not the same color, something's wrong. Hyalinosis on a PAS is usually bright pink. Sometimes it looks hyalinized, it looks glassy. Hyalinosis is the term as defined by the WSAVA group, and it means that plasma has leached or insudated into a matrix. So oftentimes we'll see hyalinosis in the glomerular basement membrane because there is, basement membrane is an extracellular matrix, and you could see hyalinosis in the mesangium as well. So I'll show you examples of that. I also use PAS to evaluate the brush border. So if you're looking at a PAS stain, first thing you need to do is start to look at all those proximal convoluted tubules. If they don't have brush borders, something has gone wrong. I know that it can be lost with autolysis as well, but I've had lots of cases that have been rotten for, or not rotten, but have been sitting for a day before they got autopsied, and they were still able to maintain their brush border. So I do think it takes a couple days of autolysis to actually lose that brush border. Use the PAS to evaluate the tubular basement membranes as well. If the tubular basement membranes are thick, then we think that that tubule is undergoing atrophy. Sometimes they'll be thick and multi-laminated, and again, it shows some type of degenerative process in the tubule or atrophic process in the tubule. And then one thing I don't have on this slide is that the GBM and TBM, so the glomerular basement membrane and tubular basement membrane, should be approximately the same thickness. If one is much thicker than the other, then there's probably a lesion. And then we, as pathologists, use the PAS to evaluate where the cells are localized. And I'm going to show you pictures of that. Next. Okay. So here is an H&E stain on the left, and these are cut at the same thickness, 3 micron for both slides. And everybody can see that the PAS is obviously darker pink. That's what a PAS does. But I think it's easier to clarify where these cells are localized in the glomerulus, because the stuff out here with this pale kind of pink wispy stuff, those are podocytes. So I know that any nucleus out here is going to be a podocyte nucleus. This is definitively a podocyte. This is definitively a podocyte. This in here with all the pink, dark pink material around it has to be a mesangial cell. So again, it just really helps the pathologist say, I know I'm looking at a lot of cells on a lot of matrix, but those cells are within a certain location. And then again, you know, pay attention to your tubular basement membranes. Is it a smooth contour, and is it a normal thickness? Okay. So trichrome, we use trichrome. It obviously mostly for the collagen staining properties of trichrome, but trichrome actually also helps us with some protein identification. So the trichrome stain is really good to evaluate glomerulosclerosis. And so now I have to do a little bit of definition of what glomerulosclerosis is, because it is often misused. Glomerulosclerosis is what happens as the glomerulus scars. And I've been asked by pathologists before, why don't you use fibrosis to talk about scarring in the glomerulus? And as discussed by MDs and reported in the human textbooks and human literature, and what I've put in Jubb and Kennedy in this version of Jubb and Kennedy, is that glomerular fibrosis is not the correct term, because fibrosis implies collagen production by fibroblasts. But glomerulosclerosis is actually just extracellular matrix. It's a whole bunch of different fibronectrins, victronectrins. There is collagen in there as well, but there's a whole bunch of other mesangial cell or mesangial matrix components, and it's predominantly produced by mesangial cells. So it's not just fibrous collagen produced by fibroblasts, it's mesangial cells making matrix. And so that's why we use this term glomerulosclerosis. And so in order to be glomerulosclerosis, you have to have effacement of capillary loops of the glomerulus by this matrix. And any of you guys that read my reports that I send out to the clinicians, I use that phrase so often of there is effacement of peripheral capillary loops by extracellular matrix that it is almost second nature for me to type it. I don't even have to look at the keyboard anymore because my fingers know where to go. So glomerulosclerosis is matrix production by mesangial cells. And then remember, even in the trichrome, the GBM and the mesangial matrix should also be the same color of blue. If you have a difference in the color of blue, then you have some problem going on with your glomerulus. Trichrome is also really good to evaluate interstitial fibrosis, and that is collagen being produced by fibroblasts, so we do call that fibrosis. If you see, if you have a couple of different processes such as immune complex deposition in your glomeruli, you can see large red dots on the glomerulus, and that's consistent with immune deposition. So it's a protein, so it stains red, and that's, a trichrome can be very useful for that. And then again, you can see this hyalinosis, which I already told you the lesion was, or the definition is plasma incidation into the matrix or into the GBM, and hyalinosis on a trichrome is usually going to be orange to peach. Trichrome actually also is really good at staining the apical brush border of the tubules, and it should stain it kind of red just because there's so many proteins labeling that, or all along that apical brush border. So you can see the apical brush border in a trichrome usually. And then sometimes in a glomerulus, it's not too frequent, but occasionally we'll see this bright red, orange fibrillary material, and that's indicative of fibrin. And so that tells us that that glomerulus has undergone necrosis. So maybe it's a vasculitis process or a glomerulonephritis, and we have necrosis of the glomeruli. Okay, so I think my residents have seen this picture so many times because it's so easy to reuse it, and I have my cheat sheet in this corner, and I lost my cheat sheet. I'm sorry, for everybody else, when I do these things in the corner, this is what a normal trichrome should look like. And so I made this slide a long time ago, and now I can't find my cheat sheet trichrome. So I keep reusing this slide over and over again. Okay, so a normal trichrome should have an endothelial cell nucleus at the base, and you should just have a blue capillary loop, and that blue capillary loop should be the same blue as the mesangium, which is right here. And if you can see on this glomerulus, you have all of these red dots all along the surface, and those are definitively diagnostic for immune complex deposition. There's nothing else that this process could be because of the pattern and their location and their frequency. If I saw one red dot, I wouldn't call it immune complex glomerulonephritis. If I saw a couple red dots, I still wouldn't call it. It has to be pretty marked and a regularly spaced is a phrase I use often, along the glomerular capillary loop. So all of those red dots are going to be the immune complexes. And then, as I said before, the mesangium is the same color as the GBM, so that's good. That's within normal limits. And then all the cytoplasm of this proximal tubular epithelial cell is bright red, and you can see the tubular basement membrane right here. This one actually doesn't show the brush border very well. I think it's kind of staining a titch blue up here. I'm not really sure why, but in general, this is a great one to say, hey, do I have interstitial fibrosis in this case? I don't have any. All of this stuff that's in between the tubules are peritubular capillaries, so that's good. Okay, Jones stain. So the Jones, and I have plenty of people email me asking, can I just do a GMS because my lab knows how to do a Gamorrean methenamine silver and they don't know how to do a Jones? A GMS is never going to work. It has to be a Jones. And it does take a while to work it up in your lab. Our lab and Texas A&M's lab does an amazing Jones stain. If you really need a Jones stain because you really want to look at a case, send it to us. It'd just be easier for us to do it for you than for you guys to try to perfect the stain in your own lab because you might use it once a year. So give us a 3-micron copy of the slide and we would be able to stain it for you. So, and that being said, if I get 100 glass slides in my box tomorrow, I'm going to be kind of depressed. Okay, so this one is a stain that it's really going to show us extracellular matrix. So the point of the Jones is to show us extracellular matrix. So it's going to stain the glomerular basement membrane black, and it's going to stain the mesangium black as well. So just like I said with the PAS, and just like I said with the trichrome, they should be the same color. If one is black and the other is gray, something is wrong. The glomerular basement membrane should be smooth. It should not have irregularities. And if we see spikes, if we see what we call holes, which I'll show you pictures of, if we see double contours, all of those things tell us that the glomerular basement membrane has been remodeled. If you have a biopsy or an autopsy specimen and there is diffuse remodeling, so all glomeruli are remodeled, then you're really worried that there is going to be underlying immune complex disease. So if you just see one glomerulus with remodeling, then don't make your entire diagnosis on one glomerulus. But if most of the glomeruli are remodeled, then you need to tell the veterinarian that it's probably going to be immune complex disease. EM would verify that, or IF could verify that. But in general, they need to know that this animal probably, if it's a living patient, needs to be immunosuppressed. And so again, mesangium should be black. If you have a gray mesangium, then you worry that there's material in the mesangium that shouldn't be there. Most likely it's going to be amyloid when you're talking about small animals. Sometimes we have very few rare cases in our system where we found other fibrillar material that was not amyloid. In humans, they have a lot of other fibrillary deposits or fibrillary processes that happen in their glomeruli, so there are a lot of things you have to do once you see gray mesangium in a human biopsy. But in a dog, in a cat, it most frequently is going to be amyloid if you have a gray mesangium. Okay, so here is the Jones stain from that. So the trichrome I showed you before, and the Jones, same glomerulus, just a different stain. So again, our cheat sheet down here is a smooth capillary loop. Mesangium is the same color as the capillary loop, okay? And if we look out in this glomerulus, we have all of these little projections, and they're all going towards the podocyte. And remember, the podocyte is an epithelial cell, so we call them subepithelial spikes. So here's another, you can see the podocyte nucleus, and you can see all these spike-like projections going towards the podocyte. And then what's a hole? A hole is basically when you have spikes that are, what those spikes are doing is they are new glomerular basement membrane material, and they're being placed next to immune complexes. So you'll have an immune complex, and then you get GBM to either side. And then eventually that GBM keeps getting placed right there, and eventually it encircles the entire immune deposit. So the hole, this little clear space, represents the immune deposit, and this is all new GBM, glomerular basement membrane material, that has encircled the immune deposit. Yes, we have a question from OSU.
Is Jones also the best thing to see mesangial interpositioning?
Yes, so the question was, can we see mesangial interpositioning with Jones? And that will be the next PowerPoint slide. You have predicted me well. Yes, so if the holes show that there's going to be immune complexes there, and then you have this remodeling towards the podocyte surface. So remember back to your physiology classes that GBM is synthesized both by the endothelial cells and the podocytes. So both cells are producing GBM, and there's a lot of turnover over the course of your life. And so in this particular instance, because it's on the subepithelial side of the capillary loop, this is podocyte production of GBM material. And so really the endothelial surface, the subendothelial surface, is quite smooth. So the endothelial cells are probably happy and healthy. They're probably not upset at all. And these immune deposits are all right below the podocyte. Podocytes don't like being in contact with immune complexes, and so they start to lay down new GBM. I like to think about it as if they're trying to wall themselves off from the immune deposit. Okay, so, and then I'm trying to see, this one right here, if everybody can see these tiny little discrete holes, what this is almost a glancing blow of a capillary loop. And so in my descriptions, when I write these kinds of cases up, I talk about en-face sections of capillary walls. So you basically have a glancing blow of this 3D structure, and you can see these holes are where the deposits are, and this is all the GBM matrix around it. The case I just read out on Friday only had these holes right on the en-face section of the capillary walls, and when I did IF that same day, I could see the immune complexes right there in that same location on immunofluorescence. So it really matched up with what I saw in histology. Okay, so now here's a silver stain of a different type of immune complex disease. Now, the previous stain, the immune complexes were subpodocytic or subepithelial or on the abluminal surface of the capillary walls. And in this disease, the immune complexes actually get deposited right below the endothelial cells. And so here's an endothelial cell, and then what happens is the endothelial cell is really upset that it's sitting next to an immune complex, so it starts to try to wall itself off as well. What you'll realize is that when endothelial cells lay down new GBM, they just lay it down circumferentially around the capillary loop, whereas when the podocytes lay down new GBM, it makes spike-like projections. So in this type of disease, when you have subendothelial deposits, you'll have what we call double contours. You have a dark region. This light region is probably exactly where those immune complexes are, and then you have a dark region. So this is what the podocyte had already been making, or what was already pre-existing, and this is new GBM that the endothelial cells started to lay down. And so they're trying to wall themselves off from whatever this material is in here, and most of the time it's the immune complexes. When I look at these kinds of cases on EM, these immune complexes for this type of disease, these subendothelial immune complexes, they're usually quite variable in size, and they can be quite large. We often talk about they become confluent with one another. So it's not like in this disease where they're discrete immune deposits sitting in between spikes. And I will show you EMs of all of these, I promise. Okay? So Any questions about the stains that I just talked about, the trichrome PAS or Jones? Anybody need to unmute themselves? I'm going to look at participants. Everybody's good? Oh, we have a lot of people attending. That's exciting.
Rachel, one quick question. You were going to talk about mesangial interpositioning. That can also cause double contouration, correct?
Oh, yeah. So I should have said that. I incorrectly spoke when I said these are immune complexes. They often are immune complexes, but in addition to immune deposit material, they are also little extensions of mesangial cells that start here. So the mesangial cells are down here, and they make these cytoplasmic projections, and it kind of dissects in between the layers of the GBM, and so that also contributes to the double contour appearance. If I see this, I wait for, I say it's consistent with mesangial interpositioning, but in actuality, when I go to EM, it's more frequent to see immune deposits there than the actual small fragments of mesangial cells. But both the mesangial cell interpositioning and immune complexes in that location can cause a double contour appearance. And remember, I think I've told at least my residents a lot that whenever you do anything to a glomerulus, especially a canine glomerulus, the mesangial cells are the first, the most common reactors, I guess I should say. So they start to proliferate and they start to send out these small cytoplasmic projections into the GBM. So, yes, we have a question here. Yep.
The top arrow at the end of that, is that one they hold?
Yeah, so perfect. So the question is, here we even have the nucleus of a mesangial cell, and he is not within mesangium proper. He's out here in the capillary loop itself. So perfect. So this is a mesangial cell that's almost started to migrate out. And, you know, we have a couple of really great pictures of EMs of that type of lesion, but I get 20 or 30 glomeruli on a biopsy that I do histology on, and then when I get EM, I might only look at one glomerulus. So being able to catch that right in the right section is really difficult. But yes, that's a good pickup, Josh. Thank you.
And Rachel,
Right,
there was a question on the chat from you, Penn, asking, are the composition of the epithelial and endothelial basement membrane, I'm guessing, is the composition of the epithelial and endothelial basement membrane different?
They are not different. So or at least not to my knowledge. So both of them are synthesizing basement membrane. The basement membrane is predominantly type 4 collagen. There is some laminins in there as well. I am not aware of ways that we have been able to detect difference in their composition. And so if you guys can remember, and I think I do have pictures later in the presentation about the trilaminar structure of the glomerular basement membrane. And so the reason it's trilaminar is you have both the epithelial cell and the endothelial cell making basement membrane, and then the middle part of it fuses to make the lamina densa. So actually, I'll just, because we're, let's see, how do I go back out? Okay, so we'll just really quickly. So this would be the EM. They will not be labeled for you on board. But if you guys can see here, there's a gray line, quite a thin line, because this is a very healthy animal. And then you have more of an electron-lucent line right below the endothelial cell, and another electron-lucent line right below this podocyte. So that's the trilaminar structure of the glomerular basement membrane. And it's because the endothelial cell and the podocyte are both synthesizing basement membrane, and they meet in the middle, and that meeting area is the lamina densa. Okay, does that help your question? I can't see the chat. I hope that was okay. Yes, thanks, perfect.
Yeah.
Okay, the next few things.
I'm sorry.
Is that okay?
Last question I had was going to be on the trichrome. You've sometimes exercised caution about saying fibrosis with trichrome in certain cases. Could you clarify that?
Yeah, so fibrosis of the interstitium, is that your question?
Yes.
Okay, so sometimes what happens is if you have acute tubular epithelial injury, you have a lot of interstitial edema, and so what happens is your tubules move away from each other. So if a resident is trying to tell me that there's acute kidney injury and we're looking at the scope together, the first thing we look at is, are the tubules back to back? When I go over to the med school, that's the first thing we ask when we show the nephrologists the biopsy. We say, are the tubules back to back? And we make them answer that question. If they say no, then we have to say, okay, is it edema fluid in there or is it collagen? And so sometimes it can actually be a mix of a little bit of collagen and a lot of edema. And so it might be important because we don't want to overinterpret scarring and say, oh my gosh, there's a whole bunch of tubulointerstitial scarring, when in fact it's just a little bit of collagen and a lot of edema. The edema can go away and the collagen would stay. So sometimes that pale blue matrix, which we don't have an example of in this picture, but pale blue matrix would be more, I guess maybe down here. It's a pale blue color, but it's not necessarily dark, dense blue collagen. So I will often exercise caution and make sure that I'm not over-calling scarring, because scarring would be irreversible and edema would be irreversible. Okay. Back to, so just a note, if you're doing a Conga-Red, please do it on an 8 micron. For any organ, it should be at least 8 microns thick. Sometimes we do 10 microns, or some labs do 10 microns. Sometimes you can have amyloid, or you can have matrix that is Conga-Red negative, and then when we do EM, it's still making fibrils. Most of the time, those fibrils are too thick. They have too thick of a diameter to actually be amyloid. So we do recognize a couple fibrillary diseases that are not amyloid in dogs and cats. In humans, they recognize a lot more. There's a lot more paraproteinemias that will polymerize into fibrils that are the wrong thickness for amyloids. They usually behave like amyloids. I shouldn't say that. Sometimes we think that some of these actually aren't as bad as amyloid. We can do an oil redo. That's going to help us identify lipid. I like to do that a lot in cats that have lipidosis in their tubules or in the interstitium. I can do an elastin to evaluate the arteries. So if I'm worried about arteriosclerosis, I'll do an elastin. I've actually started doing an elastin on most of the cat biopsies I get. If the patient presents with some type of quote-unquote Fanconi's-like syndrome, one of the differentials for that would be copper in being in the liver, also often in the kidney, and so I can do a rodenine to see if I can identify copper. So here's an example of an oil redo. I think this is an example that Dr. Conkin took from one of the cases we presented at ACVP. And all of these are foamy macrophages, and the oil redo has identified the lipid material within the foamy macrophages. And this was an example of what we talk about for glomerular lipidosis. So you have glomeruli that are effaced by foamy macrophages. This is an example of an elastin. This was actually a transplant cat that came from Penn. And we have discontinuity of the internal elastic lamina. And we have a lot of inflammation around this vessel. So I was worried about injury to this arcuate caliber blood vessel in this particular specimen. Okay. I think we're on the, yes.
A quick question by you. Oh, sorry. I think you have somebody.
Keep going, yes.
Quick question by UPenn. They ask, will tubules separate with autolysis?
So that's a really good question. Do tubules separate with autolysis? They do. I don't know to what degree they do, and that is actually going to be the summer research project that I'm going to have a vet student help me with. So I'm going to do serial biopsies of dead things to look at how much, and this is a great example of tubular separation. Like this tubule is here and this tubule is all the way over here. That's way too much interstitial matrix. So I'm going to try to do a little bit of a time course analysis, because maybe that's a better way for us to judge tubular injury than, you know, always looking at brush border. I'm going to try to quantify some of that stuff, and then I'm also going to have them biopsy the liver at the same time and do a time course analysis on that to see not exactly when, but are hepatic cords disassociated within 48 hours versus 24 hours, those kinds of things. So I'll get back to you in a year. All right. So my last one is light microscopy. It is actually not adequate. This is my myth, so that it's adequate for evaluation of proteinuric patients. So light microscopy, if you have a biopsy, especially for biopsy tissue, because you actually have a patient on the other end of that sample, If that patient is proteinuric, then light microscopy is no longer adequate for that study. Sometimes light microscopy can give you the diagnosis. If the animal has amyloidosis, you can identify that by light microscopy most often. And you can tell the clinician this animal has amyloid, there's nothing you can do for it, it's a really bad prognosis. So sometimes it can give you the answer, but oftentimes you have to go to EM or immunofluorescence in order to identify certain lesions. And then on the flip side, light microscopy is often all we need for a patient that presents just with acute kidney injury. Because in general, if they only have acute kidney injury, they have no evidence of proteinuria, then we're probably going to be able to grade that degree of injury on a light microscopy specimen. And so that's why EM and immunofluorescence are often not needed in patients that only have AKI and have no evidence of proteinuria. So, but if you have a proteinuric patient, then you at least need to offer those services. I mean, obviously it's expensive, so people aren't going to always pay for it, but you should at least say, look, light microscopy is not always sufficient for these types of patients, and you should consider doing EM and immunofluorescence. Okay, so what situations would EM and IF not be relevant for? So early immune complex GN, so if it has happened very recently and it's an acute onset of proteinuria, then we would worry that those immune complexes are so small that you're only going to be able to see them in with immunofluorescence or with electron microscopy. If there are a lot of diseases where podocytes have been injured, and so we've kind of started talking about the term focal segmental glomerulosclerosis, FSGS, and that is in the new Deb and Kennedy. And so that would be what happens when podocytes are injured and they detach, they die, and now the glomerular basement membrane doesn't have a podocyte covering, and so it will undergo sclerosis. It will scar down. There will be extracellular matrix production by mesangial cells to cause sclerosis. So that's what happens when podocytes are so bad that they die. When podocytes are reversibly injured, then you have minimal change disease, and so that means that podocytes have evidence of vacuolation, they have foot process effacement, but they haven't died. So, and that's a whole spectrum of podocyte disease processes. I will say in veterinary medicine, minimal change disease is extremely rare. It's exceedingly rare. And focal segmental glomerulosclerosis is quite common. It probably was about 25 to 30% of our diagnoses for proteinuric patients. Minimal change disease represents, less than 1% of our biopsy patients. Whereas in human medicine, there's a lot of minimal change disease in human medicine. So, so in general, spectrum of podocyte injury, sometimes you're only going to be able to qualify that or quantify that with EM and immunofluorescence. Early amyloid, we've had a couple of cases where we couldn't pick up the amyloid on the Congo red stain or in any of our special stains, and then when we got to EM, we found the amyloid fibrils. So if it's fairly recent onset of amyloidosis, you might not pick it up with the light microscopy stains. And the glomerular basement membrane anomalies, which Dr. Clark presented at ACVP this year. So there's a wide spectrum of abnormalities in the GBM, ranging from just the true ones that we've been able to identify the genetic cause for, so Alport syndrome, having a mutation in type 4 collagen, and that affects a very, we know what breeds have been associated with it, although we can find new breeds in the future. We've only seen it in dogs. I don't think it's ever been reported in cats. Mary, am I misspeaking?
Not that I'm aware of.
Okay. So, but it's also a human thing. So it's a dog and a human thing. So it could be something where we actually know that there's a mutation in type 4 collagen, or it could be something where we've just identified it in a whole bunch of dogs and we don't know the cause, like the ones that we presented at ACVP of a whole bunch of older, small breed dogs and Yorkies were predisposed. So some of those things we would need EM in order to identify these lesions. Okay. So what do I use TEM for? So it's most likely, almost always going to be due to we're going to be doing it to look at glomeruli. I have had a couple of researchers contact us looking for evidence of early ultrastructural evidence of tubular injury, but where they looked at it on histology and they could maybe only find a couple of tubules on histology that they thought might be injured. And they were wondering if they went to EM, could they find more evidence of injury? And it's difficult because sometimes certain segments of the kidney are affected, and the EM specimens that get embedded are basically 1 millimeter to 2 millimeters cubed. So it's a super small portion of the kidney that's being embedded and processed for electron microscopy. So it can be quite difficult to say, well, if I'm looking at patchy tubular injury and I might have had a micron or a millimeter cube that wasn't in that area, I would have totally missed it. So you really, patchy tubular interstitial lesions are very difficult to identify, but I've at least tried it a couple of times, and I've been successful occasionally, but it's kind of frustrating, I have to tell you. Okay, so but if we're looking at glomeruli, what are we going to do? So these are some of the things that I do like to emphasize to pathologists so that you can talk to your clinicians and guide them appropriately. So what happens is normally, wait, I think this is a typo here. So place directly into, yes. So please, I'll remove this when I send this presentation out. Okay, but normally what happens is we send out a biopsy kit, and the biopsy kit has a little vial of glutaraldehyde, and the clinician takes a core and they cut off a cube at the end and they put it in the glutaraldehyde, and it comes to us in glutaraldehyde at OSU. What also can happen is they can submit the sample entirely in formalin, and when I receive it, I can look at it in the lab and I can take it to glutaraldehyde afterwards. So I can post-fix in glutaraldehyde. In many human diagnostic labs, that's what they prefer to do. Sometimes most of the time because it makes it easier for the clinician. The clinician only has to put one sample into formalin, and then we're the ones that decide which one goes to glutaraldehyde. So oftentimes that's what it is. And we'll hopefully be able to transition to that with our veterinarians. Our veterinarians that use us are very used to having 3 vials now, and so we don't want to drop a whole bunch of change on them at once. So, but you can take samples from formalin and then transfer them to glut. The important part is that it has to be a well-fixed sample or it has to be a small sample that you placed into formalin so that formalin could fix it well. So if a clinician is out there and they're deciding like, well, maybe I might want to do EM in the future, then take your core and just put cube off a little piece, keep it in the formalin, and then we could eventually, if we decide to go to EM, take that formalin cube and put it in glut. The other thing we can do, which we're doing right now for a case that came from as a consult from Penn, is they had already looked at the paraffin block. So they had formalin fixed paraffin embedded tissue, and they had looked at the glass slide, and then they sent us the block, and we are melting down the block, and we are taking those cores from that block, and we are post-fixing it in glutaraldehyde. Those samples are good enough to diagnose immune complex deposits. You can actually see them on the formalin fixed paraffin embedded samples. They're not great they're definitely not appropriate for any kind of morphometry study. And they would be, it's difficult to evaluate podocyte characteristics on FFPE sections. But I can at least tell the clinician, hey, this is immune complex disease or it's not immune complex disease. So even if you just have a paraffin block, you can get EM from that. One thing we also did at UNC, which worked half of the time, was we would take the actual section from the glass slide, scrape it off, and process that for EM, and that would be good enough for us to identify immune complexes. So let's say, what would the situation be worth it? So maybe the biopsy was a horrible sample and the clinician only got a single glomerulus, and that made it onto your H&E slide. And all you have is that glomerulus that got processed for H&E. So you could have taken that glomerulus and say, can I identify electron dense deposits? It's probably going to be immune complex GN. Okay, in most cases, I'm only evaluating 3 glomeruli or less, or fewer, I guess I should say, since, yeah. So in most cases, it's only 1 to 3 glomeruli, so I might be making an entire diagnosis on a single glomerulus. So I have the supporting evidence from the light microscopy, and I have the supporting evidence from the immunofluorescence, but I'm only looking at very few glomeruli. What am I looking for? I'm looking for electron-dense deposits. I'm trying to evaluate the podocytes. I'm looking to see if any of them have detached, and I have denuded glomerular base membranes. I'm looking at the GBM structure. Evaluating endothelium is a little difficult because there's a lot of artifact that happens with the endothelial cells, but I'm at least trying to look at it. And then I'm also looking at mesangium, and I'm trying to find out, are there immune complex deposits in the mesangium? Am I seeing fibrils? Most of the time when I'm doing an EM study, I probably, for most cases, take 10 to 13 pictures. If it's really cool, I'll take 25 pictures. So in general, I get a couple with the capillary loops, especially if it's immune complex. I just need a couple pictures to say, hey, these are what the immune deposits look like. And then I do a couple pictures of the mesangium to say, hey, there are deposits in the mesangium or there aren't. Okay, so I already showed you this picture. Things are labeled. When I try to train residents on how to read an EM, the first thing I say is look for these little guys, which are the podocyte foot processes. If you can localize and you have intact foot processes, you know you have to be in urinary space. If you have a red blood cell, that also helps because that tells you you're most likely in the capillary lumen. And endothelial cells should be at the base. They sit on top of the mesangial matrix. They are in direct connection with the mesangium. And what happens is the GBM actually comes along here from this capillary loop. So here's your capillary loop, GBM, and it reflects and goes along the mesangium. So this region that's lining mesangium is called paramesangial glomerular base membrane. So this endothelial cell sits directly on the mesangium, and then the mesangium is also in direct contact with GBM at certain regions, okay? So sometimes I only have deposits on the paramesangial GBM. I don't know why. Maybe it's because there's an antigen here that's not on the peripheral capillary loop. But you can see it sometimes where you only have deposits out here and you have no deposits here. So I at least try to localize them for the study, or for future studies, I guess I should say. Okay, so here's a couple of cases of where EM and IF made the diagnosis. Okay, here we have an H&E stain of a 3-micron glomerulus. I have tubular injury over here, and in general, I don't really see much going on with this glomerulus. My capillary loops are open. I might have 4 nuclei here in this mesangial segment. That's pretty much all I can tell. Podocytes, I think this is podocyte here, but I really can't tell because it's an H&E stain. The trichrome stain. Again, I have trichrome being the mesangium. I have these guys in here, which I know are mesangial cells, and I have the capillary loop here, which is pale blue, the right color. All of this material here is podocyte cytoplasm. This red flocculent material is podocyte cytoplasm. I mean, this is a gorgeous glomerulus. I can see the vascular pole. I can see the macula densa. Everything's gorgeous. Podocyte cytoplasm, red flocculent material, sorry, podocyte nucleus, red flocculent material, podocyte cytoplasm. I'm at a loss to say why this patient is proteinuric. I do the silver stain. Again, my capillary loops are right here. They have a very smooth, thin contour. I don't see any irregularities. I don't see any holes. This is a gorgeous glomerulus, and I can verify that we had 20 glomeruli and they all looked like this. And then we did immunofluorescence and EM. So immunofluorescence, all of these dots are IgG, it's an antibody against IgG, and so all of those are indicative of early immune complex deposition along capillary walls. And the EM, it's a little difficult to see on the screen, at least for my students, but hopefully you guys can see that I'm outlining an electron-dense deposit here. And here's a second electron-dense deposit. This is the podocyte. So this is urinary space, and this is endothelial space, or endothelial endothelial cells and capillary lumen space. And so all of these are very small, regularly spaced subepithelial deposits. So based on the fact that I had very minimal disease on my light microscopy stains, I knew I had to do these additional studies in order to make the diagnosis. And the great thing is it's super early disease. They had no tubular interstitial scarring. They have very minimal remodeling of the glomerular basement membrane. And so this was a disease in which we can quickly interrupt the process and immunosuppress and make a really good prognosis for this patient. These patients with early ICGN, when they're immunosuppressed at an early stage, anecdotally do really well. We're waiting for the WSAVA study from the clinicians on the WSAVA study to verify what I said anecdotally. But we have plenty of examples where it does really well. We have a question over here from OSU. We have two. Ryan raised his hand, so he gets to go first. Dr. Jennings.
No, I pointed first.
I didn't see that. It's not recognized. So with immunosuppression, do these complexes ever get cleared?
So that's the question. So with immunosuppression, do these complexes ever get cleared? We know from human medicine that yes, they can be cleared because we have rebiopsy of patients with periodic bouts of lupus. So they might get cleared and then they get rebiopsied later and they get restaged or reclassified, I guess is what we use for that term, and then they can change from membranous back to mesangial alone. So yes, we know they can get removed. We have very few rebiopsies of these patients. I think the ones that jumped to my mind have been, we biopsied them and then they went in for a foreign body later because it was a Labrador, and they still had some immune complexes, but it was not nearly as severe as the previous one. But clinically, their UPCs tend to go down, and their clinical signs resolve. If they have nephrotic syndrome, it can go away. We have another question from Dr. Permandanan.
So what's wrong with the podocyte?
Okay, sorry, I should have mentioned this. So what's wrong with the podocyte? So if you are looking at an EM on boards, one thing you should always include in your description is the quality of the foot processes. And so when I showed you this image, I said, look for these little guys. These are little foot processes. You cannot see them on this particular image because they have complete global effacement. So all the foot processes have fused into this huge glob of podocyte cytoplasm. These complexes are very irritating to the podocyte, and the podocyte cytoskeleton seems to fall apart when they're in contact with the processes. And then the other thing that happens is, in addition to fusion of the foot processes, is you get these little tiny cytoplasmic extensions, which are called microvillus transformation. And this happens probably in about a large proportion of our cases where you see podocyte injury. It's not always due to immune complexes. And to be clear, you have foot process effacement in many diseases. It's not just immune complex disease. Foot process effacement happens in pretty much any time a podocyte has any kind of damage. So the microvillus transformation, some people like to say, well, maybe it's trying to make new foot processes and it's not really good at it and it's making on the wrong side of the cell. That's probably not true, but it's a good way to think of it, right? Like he can't maintain his regular foot processes, so he's getting these little cytoplasmic extensions. But it's indicative of podocyte injury. So minimal change disease, FSGS, they will have the exact same podocyte foot process effacement, and many of them have cytoplasmic transformation of their, microvillus transformation of their cytoplasm. Okay?
Wait, Rachel, it may just be my screen, but I now have a fairly big gray / box in the middle. Do you see that at all?
I don't. Does anybody else have that? Chat?
Hopefully not. Yes, okay. Yeah,
Other people are.
Everyone else's too, and it just appeared maybe a couple minutes ago.
Even when I changed my slide?
Yes.
Okay, well.
Okay, it went away.
Okay, well, let's see. We'll just try it from this way. And I'll try to make it big again. We'll see.
Yeah.
Okay.
Yep, great.
So we have about 5 minutes to get through the last case. And so this is another one where we definitely have abnormalities on the glomerulus. We have an adhesion between the tuft and Bowman's capsule, and we have this extracellular matrix, so we have segmental sclerosis and a synechia. So we have an abnormal glomerulus. The PAS, again, shows the same features I just described, a little bit of mesangial hypercellularity, but if I'm looking at the glomerular basement membrane, it looks pretty good. This one doesn't look as good. It looks a little thickened. This one looks a little wrinkled. So I've got lesions, I just don't know what caused them. Same thing with the trichrome, and I showed you before where I said you're going to have, if hyalinosis happens on a trichrome, it's going to be orange to peach, and so this is a situation where it's orange. But again, a pretty broad adhesion and effacement of capillary loop, synechia and sclerosis. And then I did EM. I also did IF. The IF had negative staining, so I didn't take any pictures because it was just a black screen. But the EM shows glomerular basement membrane here, and here, and here, and all of our podocyte foot processes are effaced. So we have global effacement of podocyte foot processes. And we have this microvillus transformation of the podocyte cytoplasm. And if we're getting a close-up view of this glomerular basement membrane, it's thickened, but there are no electron-dense deposits in this case. There is no evidence of immune complex deposition, either by IF or by EM. So I can tell the clinician, the reason your dog has proteinuria is your podocytes are injured. I don't know exactly what caused the podocyte injury, but I can definitively tell you it wasn't immune complexes. So this is a non-immune complex mediated disease. Immunosuppression is not warranted. It would not help the patient. So this is a situation where we really had to, based on this, say, I see a lesion, I see tiny irregularities in the glomerular basement membrane. This is probably an area that was a thickened capillary wall to match my thickened capillary wall out here. But I don't know why. Hopefully, once we identify a great subset of these cases and we have pedigrees and we do GWAS, we can find causes of this podocyte injury. We have evidence that's already been effective, at least not effective, but has already been published in soft-coated Wheaten Terriers, looking at GWAS and proteinuric soft-coated Wheaten Terriers, and we were able to identify 2 podocyte genes that had mutations in them that tracked with the heritability of the proteinuria.
In humans, is there any effective treatment for something like that?
So the question is, in humans, do you attempt to treat this? So there are, in humans, we recognize primary podocyte injury and secondary. And they use those terms incorrectly. So primary to them means idiopathic, means we don't know. Could it be a gene? Yes. Could it be obesity? Yes. Could it be hypertension? Yes. Once they identify a gene, it goes into the secondary category. I hate that. But what that means is if they know the patient's hypertensive and it has this, they control the hypertension. If they know the patient's obese and has this, they tell you to lose weight. So they hope to modify some of the environmental characteristics that are going to decrease the amount of stress your podocyte sees. But this is irreversible, so it might always have some degree of podocyte injury. Okay, here are just other pretty examples of podocyte injury. Here's a podocyte here that has these large cytoplasmic vacuoles. There's a lot of research now about autophagy in podocytes and how the podocyte should be extremely autophagic. It should completely be always turning over its cellular machinery because as we think about it, it's pretty much terminally differentiated. It's very hard to get a new podocyte. So they have a very high autophagic flux. Autophagic flux? Yeah, that's how you say that. So then in proximal tubular epithelial cells do a lot of autophagy, and when you start to slow the rate of autophagy, they are more likely to detach in rodent models, and they are more likely to result in segmental sclerosis in rodent models. And some of those things have been applied, some of those same antibodies to look at presence or absence of autophagic vacuoles have been applied to human renal biopsy material, and they have seen a decrease in autophagy of human renal biopsy material with this diagnosis. So it's probably the new, hey, let's try to promote autophagy as a new drug. And here's a really cool one. So this is the capillary loop. This is, I mean, it was a messed up glomerulus to begin with because we've got like 3 or 4 mesangial cells, which is abnormal on an EM specimen. But here's one podocyte, and he has this huge vacuole. Sometimes we call this even a pseudocyst. And this one, I mean, this is all material that is just encapsulated and encircled by this podocyte. And we even have connection between this podocyte and Bowman's capsule. So there is some discussion about whether or not these podocytes can be part of the parietal epithelium, whether they can interact with the parietal epithelium, whether they can transition from visceral epithelium to parietal epithelium. Obviously, lots of research needs to be done, but I think it's a pretty picture. And then as Dr. Clark presented at ACVP, you can have abnormalities in your glomerular basement membrane. So this particular dog is a very small breed dog, and you have all these different laminations of your glomerular basement membrane. It is not just a dense, normal, uniform, characteristic GBM. It has multi-laminations, and these dogs are proteinuric, and they don't have evidence of underlying immune complex deposition.
In the vacuole, do you recall seeing this thing light microscopically?
So the question is, in this particular case, would I have seen this light microscopically? I saw really abnormal glomerular changes, and I can sometimes pick up the fact that they have large vacuoles like this on H&E. I've never tried to say if I see it on EM, do I definitively see it on H&E? I do have a tall blue section of this exact glomerulus, so I could pick it up on that exact glomerulus in the tall blue section. Just more glomerular basement membrane lesions that have no evidence of electron-dense deposits, and the IFs were completely negative. So again, you can have abnormal GBMs, and that can cause podocyte, or it might be associated with podocyte damage. It might be due to podocyte damage, because as we said, the podocyte makes the GBM. And so some of these things might be, it could be a vicious cycle as well. Okay. Quickly for IF, immunofluorescence, these are the ones I label. I label heavy chains, I label light chains, and I label parts of the complement pathway. C1q never works, but we do it anyway. I have to use fresh frozen tissue, and it's got to be OCT embedded. And I compared my immunostaining to immunohistochemical labeling. So formalin fixed paraffin embedded sections of the same case. So I knew it was IF positive. Could I see a similar staining pattern on IHC? And it was awful. So here would be an example of this. We've got a great granular pattern along capillary loops in this glomerulus. I know it's IgG. I have great positivity. And then this is a different case. Sorry, this is a different case where I knew this was a case of podocyte injury. This was not immune complex disease, and everything is staining positive with immunohistochemistry. We're still trying to tweak this to see if we can get a better granular staining pattern, because it would be wonderful if we didn't have to use fresh, frozen, OCT embedded specimens. But I can tell you, we've tried multiple different preparations, we've tried multiple different types of incubations, and we've tried multiple types of antibodies, and none of them have seemed to be working. And in human medicine, they don't trust this, they only trust the IF. Unless you're from Europe, apparently they trust it in Europe. Okay. Yay, I did this wow, I'm very, very excited with my timing. I know I rushed at the end, but did you guys have any questions for chats? Yes, yes, yes. Okay, that was for the big gray box. We have to clear out of here because we have another set of rounds in 3 minutes. If you guys have questions, please email me. I will post this presentation, and I will also give you a PDF of most of these slides, and that will also go on Dropbox. Thank you guys for attending, and I will get in touch with you early next January. Hopefully we'll be booking Dr. Goldschmidt for mammary gland neoplasia. He promised me at ACVP, so I'm going to hold him to it. Thanks, guys.
Thanks, Rachel.
Thank you, Mary.
Thanks. I'll stop recording now.